We are talking about reads coming together in genome assembly and making genome assembly the standard in genomics. However, it's also a call to the genomics community coming together and developing the right tools that we need to be able to do this. First, I just would like to say thank you to the entire applications team. As Dan mentioned, we are a global team and work in many different areas, and most of the people from this team have been part of making this talk and doing all the wonderful things that they've done in the past years. So thanks to all of you guys, and you are really the best team. All right, when we think about haplotype resolved genetic variation detection, we usually do it in a way where we use an alignment-based approach. Now, what we mean by that is we have a reference, we align the reads to the reference, and we can call all the different variants. So here we're showing sort of a made-up example where aligning to this reference, we can call small variants, structural variants, and modifications. And that works really well for a big part of the genome. An example of that is here, we're looking at a gene that has a pathogenic SNP on one side, and it has a pathogenic insertion on the other side. And because we have long reads, we can phase, and each haplotype, and we can see that this is a compound heterozygote because they're on different alleles, and we have a non-functioning protein from each. So that works well in many cases. However, there are some cases where mapping to a reference doesn't work as well. For instance, in immunology, we have the IHG region, where it is highly polymorphic, and every person is almost different. So there's so much going on here that if you just use a standard reference, you can't align reads to it. An example of that, or an example of how to overcome that, is we did a project where we looked at antibodies from single cells, and instead of mapping back to a reference, we did a de novo assembly from the same person on his IGH region, and we were able to map the antibodies back to the own person's personal genome. This work has just been published, and John Balaurier was one of the main authors on that. John isn't with us anymore. He passed away earlier this year, so just acknowledging his part in this. OK, so we just saw that assembly can be really helpful when you're trying to map something that is very complex. So why isn't assembly used in everything? Why don't we do it all the time? And the difficulty or the challenge with assembly is that you're really trying to assemble two or more things that are very similar and try to map them or assemble them individually. So for humans, when we think about the human genome, we think about maybe think about the diploid 3 megabases or 3 gigabases. But really, it is a 6 gigabase. You have two haplotypes, and so the problem is bigger because they're so similar. In microbial assembly, if we think about cultured isolate sequencing, we do that really well. The ML teams have made great models. We can really easily assemble isolates. But actually, only 2% of all bacteria can be cultured. So the system is more like this in a microbial or a metagenomic sample. There's many different things, and many of them can be very similar, with strains only being different in very few places. So to help tackle all of these problems, in the last year or so, we've really had the community bring some amazing tools out. And really in the last year, these 4 tools that we mentioned here have just changed the way that we can do genome assembly with nanopore sequencing. And really, that's taken us from being sort of a dark horse in assembly to being the new gold standard. But don't just take my word for it. Sean is going to tell you exactly how that works.
Thank you so much, Sissel. Now, Sissel has just made an extraordinary claim that nanopore sequencing is now offering the gold standard for de novo assembly. Well, I'm here with the receipts, and I've got about 10 minutes, so let's dig in. We'll start with prokaryotes. So these are bacteria and archaea. And as Sissel mentioned, prokaryotic genomes individually are relatively small and easy to assemble. They've been a strength of nanopore sequencing for quite some time. While they have enough repeats to confound short read assemblies, these tend to be trivially spannable by long reads, allowing us to build circular chromosomes that completely represent the genome of a prokaryote, as well as recover any plasmids. Even here, though, we're seeing constant improvements. For instance, last year, many internal teams worked to put together our no-miss protocol, an end-to-end solution for isolate sequencing. Furthermore, a recent assembler published by Ryan Wick, AutoCycler, makes the bioinformatics even more robust for isolate assembly. Finally, again, as Sissel mentioned, hard work from our ML team has led to constant improvements in raw consensus accuracy, with now the majority of bacterial species being assembled at over Q50 consensus accuracy genomes. But, as mentioned, only a small fraction of microbes can be cultured and studied as isolates. For the rest, we need to take a metagenomic approach. This is where we sequence the DNA of all of the organisms in an environmental sample and try to tease them apart bioinformatically. Now, this last year has seen a step change in our ability to do this with a new nano-MDBG tool from Gaetan Benoit and colleagues. To benchmark this new assembler, we sequenced the Zymobiomics fecal reference sample, a real human fecal sample that's been cultured to a large amount and aliquoted for reproducible study. So we sequenced this on 2 of our PromethION flow cells, and each of these achieved almost 200 gigabases of sequencing output. This was assembled and then binned into over multiple hundreds of high-quality metagenome assembled genomes, or MAGs, as well as hundreds more of medium quality. The more data we used and assembled, the more MAGs we got, but we did see that with one whole flow cell, we were able to assemble about over 90% of the mid to high abundance species in the sample. Furthermore, we find that we are an extremely attractive value proposition relative to other platforms. Full details of this can be found in our apps note that we just released, available via the QR code on the screen. Now, fecal samples actually are not the most complex microbial communities, though not by a long shot. So to really put this tool through its paces, we sequenced a compost sample, generating almost 1.5 terabases of sequencing data over 8 PromethION flow cells, all of them coming in north of 150 gigabases of output. These assembled and binned into over 5,000 MAGs, smashing the record for the most MAGs from a single sample. Most of these species had never been sequenced before. They were not in the genome taxonomy database, nor did they overlap with 2 recent large soil MAG studies, although you can see those databases also don't overlap with each other. So it really is true that soil tends to harbor an immense amount of novel biodiversity. From this, we were able to identify and characterize tens of thousands of antimicrobial resistance genes, biosynthetic gene clusters, and metabolic enzymes, providing rich information about the biology of this sample. All of this data will be released shortly, but you can find more information about it on our apps poster, out in the hallway, or online via the QR code on this slide. Turning to eukaryotes. Now, I'm about to get really excited about human de novo assembly, so I want to start with a slide that caveats that most of the time you do not need de novo assembly. Our alignment-based approaches, like you can get with our WF human variation pipeline from our epitome team, they're extremely powerful, providing you small nucleotide variants, structural variants, and mod frequencies, all phasable to local haplotypes. This information richness can yield deeper insights into the biology of your sample relative to short-read approaches. And a great example of this was a talk by Wendy Chung from the Beacon Consortium, highlighting increased diagnostic yield from long-read approaches. Encourage you to check out her talk, available at the QR code on this slide. If you do need to do de novo assembly, however, Nanopore is absolutely the answer. At London Calling last year, we unveiled what was then the most contiguous automated assembly ever. And this is, year on, our update, our new best automated assembly ever, using the same recipe, our nanopore-only telomere-to-telomere sequencing recipe of 2 ultra-long flow cells plus 1 flow cell of pore C data used for long-range phasing. And for this sample, the HG01243 sample, we were able to assemble 34 of 36 non-acrocentric chromosomes as gapless sequence spanning from one telomere to the next. The remaining chromosomes were all assembled as T-to-T scaffolds with a maximum of 2 gaps. The assembly size and other metrics all look amazing. Now, while this is the best of the best in the assemblies we've produced, it is not an outlier. And to demonstrate this, we compare nanopore-only assemblies to the recently released HPRC Human Pangenome Reference Consortium Freeze-2 hybrid genomes. Now, this is an incredible data set just published of hundreds of genomes assembled with what was last year the absolute gold standard for de novo assembly, a hybrid approach utilizing both HiFi and Oxford Nanopore ultra-long reads, as well as long-range phasing information. So they applied this to hundreds of samples and got a median of 15 telomere-to-telomere contigs across all of these assemblies. However, when we compare to 10 samples that we've sequenced with the nanopore-only recipe, we see that with less ultra-long data and no HiFi data, we're getting almost twice as many telomere-to-telomere contigs. Less data, more contigs that are telomere-to-telomere. What more could you possibly want? Well, you could want all of this faster. Enter HiFi ASM ONT. So this is a new tool from Haiyue Cheng and Hing Li. Haiyue spoke yesterday at our Bioinformatics breakout session. If you missed it, I encourage you to go back and watch it on the virtual platform. In brief, though, this software provides a 4x speedup over Dorado Correct plus Verco, our other telomere-to-telomere assembly pipeline, while not requiring any GPU. Furthermore, it works with both ligation sequencing data as well as our ultra-long sequencing data. We see that when we use our ultra-long sequencing data, we get assemblies of roughly comparable contiguity to our Dorado Correct plus Verco pipeline. With our ligation sequencing data, which is shorter but has lower input requirements and has slightly higher yield, we do see slightly less contiguous assemblies, and we see some local misassemblies in centromeric regions. These are still, however, better assemblies than you can get from any other single platform out there. Because of the lower input requirements for our ligation sequencing, we're also able to apply this to a broad diversity of samples, and we find that HiFi ASM ONT is building amazing genomes for plant and animal samples. Some considerations to bear in mind while using this, because it is a very flexible platform. Especially what I want to highlight is that care should be taken in interpreting outputs if you don't have long-range phasing information. So if you have either Hi-C, Poor-C, or trio sequencing data, both HiFi ASM and Verco will build what are called haplotype-resolved assemblies, where they use this data to connect sequence from maternal or paternal haplotypes on either side of long runs of homozygosity. If you don't have long-range phasing data, however, HiFi ASM does something interesting. It still builds extremely contiguous sequences, but on either side of the runs of homozygosity, it picks a haplotype at random. So this means you build pseudo-haplotypes that can be stretching from telomere to telomere. However, they do not represent exact maternal or paternal haplotypes. This is amazing for pangenome works and building catalogs of variants, but just something to bear in mind, you still do need long-range phasing data for haplotype-resolved assemblies. But then HiFi ASM will use that data and build these beautiful assemblies. All right, turning now to using these assemblies and what we can do with this to unlock new biology. So we're starting with a sample that we sequenced in collaboration with the NYU Undiagnosed Disease Program with the mission of applying the latest genomic technologies to end the diagnostic odyssey of families with undiagnosed genetic diseases. So this is a sample with developmental delay and regression and a host of related symptoms. They had a variety of different genetic tests done, including a karyotype that showed a translocation of unknown significance between chromosome 2 and chromosome 16, as well as a host of other undiagnostic tests. Now, the UDP was able to reanalyze their short-read WGS data and find a collection of breakpoints, but they couldn't build the whole picture of what this rearrangement looked like. They just saw this scattered collection of breakpoints, some of which involved chromosome 2 and 16. Long-read, alignment-based approaches were able to provide more context, linking together multiple of these structural variants, but they still couldn't build the complete picture of the rearrangement. So we used our T-to-T assembly approach, building most of the chromosomes as T-to-T contigs, including these two, which we could map back to chromosome 16 and chromosome 2, and very easily see this translocation event within our de novo assembly. Furthermore, though, if we zoom in, we can see at the translocation break-ins, we don't have a clean translocation. Rather, we seem to have some sort of local chromothripsis event where we have a shattering into multiple small fragments that all get rearranged relative to each other. And this is all cleanly represented in our alignment of our T-to-T contigs back to the reference. We can see that one of these disrupts an important gene for neurodevelopment, BCL11A. And so this is a likely causal variant for the symptoms that we're looking at. Okay, turning to another example of a use for de novo assembly, resolving clinically relevant regions that are challenging with reference-based approaches. Specifically, we're looking at the SMA locus. So SMA is an autosomal recessive disorder caused by a loss of function of the SMN1 gene. Now, diagnosis and carrier screening is made challenging by the fact that this is a highly complex and highly variable segmental duplication in the human genome with a very closely related pseudogene SMN2. Alignment-based approaches can show copy numbers for both SMN1 and SMN2 and identify carriers that only have one functional copy of SMN1. However, there's a class of carrier known as a silent carrier where a duplication of SMN1 on one haplotype masks a deletion on the other. Right now, there are some marker SNPs that can indicate that you might be a silent carrier, but the only way to definitively resolve this without assembly is extended pedigree studies. So we surveyed the Coriell database for some cell lines that had marker SNPs indicating they might be silent carriers. And this is an example right here containing both of the most highly associated markers. However, when we sequenced this with our T-to-T recipe, we were able to fully resolve the SMA locus in both haplotypes and see that both haplotypes contained a functional copy of SMN1, eliminating the chance that this individual would be a silent carrier, even though our best marker approaches would indicate that they were. All right, so I hope I've convinced you now that Nanopore is providing the gold standard in de novo assembly and showed you some interesting use cases where this can unlock novel biology in your sample. To talk a bit about where we're going next with de novo assembly, I'd like to welcome back Sissel.
Thank you. So I get to do another fun thing at the end, which is talk about what we think is coming. So this is just a recap of what Sean showed. On the one hand, we have the best of the best T-to-T ever produced, but on 3 flow cells. And on the other hand, we have partially phased near T-to-T, but only using 1 flow cell. So the question is, can we combine the two? What we did is we took some of the flow cells that Rosemary talked about yesterday, so the higher output flow cells that are coming. We had a bit of an early access to them. And we put a mix of 2 libraries on there. We put a normal LSK library and then some POR-C data sample as well. And this is what it looks like when we run that over time. What you can see is that we ended up getting over 40x of around 43 KB and 50 LSK data, and over 10x of POR-C from a single flow cell. And the question is, how does this assemble? Well, it turns out it assembles very well. We got a fully phased T-to-T assembly from 1 flow cell. I just want to say that LACMEL actually, at the tech update at NCM in 2024, actually said that this would be possible soon. So I'm very happy that we did it. But let me just repeat what this is. On a single flow cell, with a combined set of data inputs, we were able to get a fully phased T-to-T assembly that can be used for many things, obviously. But this is really a paradigm shift, and we hope that moving forward, this will be the new standard. So in terms of a summary, I hope we've convinced you that, 1, you can do assemblies to do a lot of the things that you would normally do with alignment-based approaches. This is true for both things like rare diseases, like Sean showed, and PopGen. It's true for plant and animals. We put a little hamster on there because we can also use it for things like in biopharma, like the CHO cell line, which isn't always what you think it might be. On the prokaryotic side, we can use it for isolate sequencing and, of course, for metagenomic sequencing. And then the second thing I'd like you to take away is that you should really be doing your assemblies with nanopore sequencing because it just works really well.